And so good afternoon, everyone, or good morning for someone. Welcome to join the GW for Water Security Alliance webinar today. Today, we are very glad to invite Professor Barbara Kasprzyk-Hordern, and she's also the co-director of Water Innovations Research Center at the Bath University, and she's been working in this water and the public health areas quite a lot. So today she's going to share with her recent insight regarding the wastewater-based epidemiology and the One Health. So she will give the talk today. So if you have any questions during her talk, you are more than welcome to put in the chat. And then after her talk, you can also ask her personally about your question, or she will answer the question you put in the chat. So we would like to start here. So let's welcome Barbara.
Thank you. Thank you for a nice introduction, and thank you for inviting me to give this talk. I'm going to share. I hope I will succeed. Okay. Please let me know if everything works fine.
Looks perfect.
Yes. Great.
Okay, I'm going to remove everything that I can from the screen. I have managed to learn all the tricks, and I'll choose this nice laser pointer. Okay, good. So yes, I'm going to talk about wastewater-based epidemiology, and in a bit wider context of One Health. And I'll possibly pose more questions than answers in terms of how wastewater-based epidemiology can be used in One Health concept. This talk is work of many people, so I'm talking about there are many people working to make sure that there's data that we can share with others. So I would like to thank our Environmental Chemistry Public Health Research Group, and also our excellent collaborator, Wastewater, the European SCORE group that I've been part of from the very beginnings, over 10 years ago now, 11 years ago now, when we started working within the field of WEB, and various other collaborators, and also people present here today, so people within our Water Security Alliance. So I will start with a quick definition of what wastewater-based epidemiology is about. Maybe not the definition, but just an introduction to the concept. So we are constantly exposed to various different stresses, as we tend to call various different chemical, biological, physical agents that might be affecting our health. And here they are, and as you can see, SARS-CoV-2 is one of them. And if we are exposed, we will respond to this exposure in various different ways, and it can be via changes at the genetic level. We might see formation of various different proteins. We still be able to capture the pathogenic organisms, and what chemists like is we will see changes in the metabolic level. So if any potent toxin, any chemical, foreign chemical, we call it xenobiotic chemical, enters our bodies, is going to be metabolized very quickly if our body is capable of doing so. And the role of metabolism is simply to make the chemicals that we're exposed to more polar, so more likely to be excreted with urine. And wastewater-based epidemiology kind of builds on this. So all the various different markers, biological, chemical, physical, exogenous, so the ones that were present out there in the environment to which we're exposed to, and also endogenous, so the one that works formed in our body, they're all excreted via urine if they're more polar, or feces if they're less polar. But they all end up in our sewage system, and with the sewage system, they end up in our treatment works. And historically, we used to focus on wastewater treatment plants. We collected samples, and we still do it, collected samples that are representative for the whole community contributing to treatment works. And you all know that it can be 10,000 people, it can be also 2 million people contributing to one wastewater sample. and nice picture here. But with SARS-CoV-2 work, there's been a lot of work that moved a bit closer to at source applications. So looking at, for example, hospitals, schools, and other areas. But today I'm going to talk about wastewater treatment plants. So covering larger population, because I see there is a benefit from doing it, mainly from the point of view of early warning systems. So yes, wastewater is a fingerprint of communities' health and lifestyle, and the concept is extremely simple. We all agree, but methodology is very sophisticated. So we're looking at tiny little quantities of PPT levels, parts per trillion levels of chemical agents in very complex matrix such as wastewater. So in our group, we actually spend most of our time developing methods, finding biomarkers and then developing methods. And I like showing this slide because it summarizes what we do. So urine samples, and actually urine and feces samples now, from when thinking about SARS-CoV-2 work, from thousands of people would have to be tested to verify community-wide health status. This is expensive and logistically impractical. Actually, a lot of expense has gone into SARS-CoV-2 test and trace, for example. But only one wastewater sample is needed to assess communities' health with high certainty, low cost, and real time. Again, there are massive limitations to WB, and I could never say that WB is going to solve all our problems. It's a diagnostic medium for community-wide health status assessments when we look at wastewater. But we can't identify individuals. We can't differentiate between sex, age, co-exposure of various different chemicals. So I always look at wastewater-based epidemiology as one tool in a toolkit, and it has to be run alongside other epidemiology tools. And it should more identify problems and issues in catchments, the communities, where and trigger the kind of individual testing. And I see this as a future for WBE, and it actually aligns very nicely with One Health, and I will talk about it in a second. But before I do this, I want to show this complicated slide. I always show it because it tells us about what we do in WB to make a success of it in a quantitative way. So, and I always use cocaine as an example simply because it's one of the first markers that we looked at several years ago. So it's well over, well, I think it's around 14 years ago, the first studies came out. Cocaine can be consumed and can be also directly disposed of. And this happens to basically all agents that we are looking at in this context. And this always gave us a headache because we don't want to capture this direct disposal. We want to capture what we were exposed to internally. So to differentiate between the two, we need to look into indigenously formed markers. And these are metabolites in the case of cocaine. As you can see, various different metabolites are formed, and actually each of them will be telling us different stories. So this is the kind of benzoylecgonins is produced, actually is the most common metabolite used, very much linked with the oral administration. This one is produced with smoking, and this one is by co-administration with alcohol. So by looking at those in wastewater, we can back calculate exposure, but it's not as easy as I say. In order to do this, we need to understand metabolism pathways for individual chemicals. We have to understand population size that provided the sample. We need to capture the flow of wastewater to normalize to flows, variable flows. This is the biggest variability in our measurements. And also, we need to have an understanding of biomarker stability. So it's not good to say, okay, I'm going to focus on benzoylecgonin because it's a characteristic biomarker for human exposure. We need to know that benzoylecgonin is stable in the sewage system and also during sampling. So we do evaluate stability of our markers. And once we have it all, we can then plot it nicely and show changes, spatiotemporal changes locally, nationally, internationally. And here we have data that's produced by SCORE group that feeds into the to the European Monitoring Center for Drugs and Drug Addiction Early Warning Systems. If you're interested, you can have a look annually. The SCORE group, we are a member of SCORE group, feeds in the data to EMCDDA, which is wonderful, which is amazing. And this was our first real application of the work that we've done. That's why I always come back to this. And I never want to forget people that I emphasize who really started this. And it was Christian Dalton. He's retired now, but he used to work for US European Protection Agency. And he gave this idea. He suggested that it's if we see biomarkers of various different illicit drugs in wastewater, that might indicate exposure. First study was produced in 2005, and in 2010 we had our SCORE group working across Europe, trying to collect data, working closely with the EMCDDA. And afterwards we received funding from various funding bodies, and I want to always acknowledge and appreciate the European Commission for having trust in us, really, because in early days no one really believed that wastewater can be used for this purpose. So lots of funding for our European network, and also lots of funding recently in the UK. So I'm very thankful for this on behalf of everyone who is involved in WBE, really. So we started 2 years ago with illicit drugs and alcohol. Then we moved on into food toxicants, infectious disease, and NCDs. And you can see SARS-CoV-2 is here. And many people will know about WBE because of SARS-CoV-2 work. But there are many years of trying to make it work before SARS-CoV-2. And today I will talk about everything but SARS-CoV-2, because I feel that some of the chemistry dimension, the quantification that we apply, is the one that we need to focus on. Moving on to our group, Environmental Chemistry Public Health Research Group in Bath. As I mentioned earlier on, we are focusing on various different biomarkers. So we try to select them, validate them, and also introduce new analytical approaches that enable quantification of our markers in a very complex matrix. And we are focusing on both community-wide disease status assessment as well as exposure to environmental and food stresses. And these are various groups of biomarkers we are looking at. So from hazard indicators such as the stresses themselves, so we have pesticides, lifestyle chemicals, through to exposure indicators, so metabolites of markers of exposure, and health outcome indicators. So these are these indigenously formed markers that can inform, for example, oxidative stress or inflammation status in our bodies. And most of our work actually has gone into intervention indicators, which are pharmaceuticals, because this is the easiest that we can do. Some of the markers are really difficult to capture, especially those that are of larger size, such as proteins, so the group 3 and 4. And we applied our approaches in various different dimensions, and some areas are captured here. So we've been working in the development of early warning systems, also to assess public health status, exposure studies, and also to evaluate interventions. And I will touch upon this today. But before I start, I wanted to emphasize one thing. Wastewater-based epidemiology, historically and still now, is focused on us, on communities. So we think about how people are exposed to various different stresses and what it means in terms of public health. But there is really much more to it, and I wanted to touch upon this today. So I wanted to emphasize this One Health dimension. Instead of looking at individuals, the communities, we should be looking into catchments. And today I am going to focus on one particular catchment, the Avon catchment in south west England, where we have done a lot of work. So well over 10 years worth of work. So Wastewater-based epidemiology, I see it as an enabler of One Health, and I think all of you know what One Health is about, but definition is provided here. 4 years a cross-sectoral, multidisciplinary efforts aimed at holistic understanding of management of public and environmental health. And One Health has been applied successfully. Some people might disagree in tackling one of the key challenges that we're facing now, which is antimicrobial resistance. And I feel much more can be done in this context. So why is that? Wastewater-based epidemiology can evaluate environmental public health status, but also inform One Health actions, and very important, evaluate mitigation strategies. Very often we hear that some of the interventions at the community level were successful, and when I ask, how do you know? Do you have evidence for this? I get an answer, well, it's not possible to capture the success in a quantitative way. And I feel that WB can capture some of the successes and actually some of the limitations of public health interventions. So I will mainly touch upon human population as a key driver of biochemical burden in the intercity system today. And I'll just say a few words about how we try to use pharmaceuticals as a proxy for community-wide health, and also WB as an early warning system for chemical exposure. So I mentioned earlier on that we focus mainly on the Avon catchment, or in this talk I will focus on it, but obviously we do so much work with the other groups that are in Europe and internationally. But today it will be only about our catchment, and most of you know where will be able to find yourself on this map. Just to simplify, this is where Bath is, this is Bristol. What we have done here, we looked at, and this is the Avon catchment, the Avon River itself. So what we looked at here, we tried to capture the whole, as many people as possible contributing to this capture. So we targeted 5 towns and cities. And here, for simplicity reason, I would just call them all cities, but obviously town A is too small to be called city, and town B too. But just for simplicity reasons. So we looked at 5 treatment works because they serve these 5 towns and cities. We also looked at river water before and after discharge points. So these are the points S1, green color indicates before discharge points, on wastewater, S2 is after discharge points. And these 5 treatment works contribute to capture wastewater from 75% of the overall population in the catchment, which is really handy in terms of trying to understand the burden from communities in this catchment. And just for a brief explanation of what these colors mean. So grey color indicates wastewater treatment plants that serve only communities without any industrial input. So it's communal wastewater, while orange color indicates communities that have larger contribution from industrial wastewater, and it's in this case will be 30% or higher. And so in this particular talk today, I will focus on various different chemicals and also biological agents, as you can see here, and probably can't read it. I can hardly read it, but I will just briefly introduce you. The yellowish orange color is these are pharmaceuticals, various different pharmaceuticals, from antibiotics, antifungal agents, through to anti-cancer, diabetics, antidepressants, and analgesics, so various different groups of pharmaceuticals. We have also personal care products and some industrial chemicals such as plasticizers, UV filters. We have also lifestyle chemicals, so caffeine, nicotine, and the metabolites, as well as some of the illicit drugs. We have also traditional water quality indicators, so ammonia, COD, nitrogen, phosphorus compounds, and we also have resistance genes, just to touch upon AMR. And we worked with them all together. There are various different methods used to capture them, but we have managed to, this is work of many people. We looked at what they tell us, really, when we look at them holistically and in the context of catchment, and not necessarily individual treatment works. And this is a typical example of the work that we produce. So we have here a pie chart showing various different classes of chemicals, so lifestyle chemicals, anti-inflammatories, and antidiabetics. You can see a very high percentage of those. And this is in wastewater influent. And information that is captured here, we don't see much. You have groups of chemicals, but each group of chemicals will be telling us different stories about what communities do and whether they have some problems with health, because we can use pharmaceuticals as proxy for health issues. So wastewater influent tells about public health. Now, when we look at the catchment, so the river water, and here we have, this is the average for the whole catchment, and it's interesting how similar the distribution is across various different locations. This pie chart and these different colors capture information about environmental health, and I would like to see more of integration. So WB does one thing, and those interested in environment do other things. We need to really integrate this to provide this very thorough grounds for One Health thinking, these are the kind of the general results and what we have observed in this particular catchment. So, for example, we managed to calculate the overall burden from CECs, so chemicals of emerging concern, in this catchment. So 141 chemicals, it's about 170 kilograms per day of chemicals of this type we introduced into the catchment. And most of them sit in the liquid phase, very few in the solid phase, but those very few in solid phase actually are very important to us. Some of them are antibiotics, for example, and fungal agents in particular. And some of the after antidepressants will sit into solid phase. Most of them get removed, which is great news. That means that our treatment work very well. But some still remain, and you might say 14 kilograms a day of CECs, it's not a big issue. Actually, it depends on what sits in this 14 kilograms. And we, for example, have evidence now that antibiotics in our catchment might be causing us a bit of a headache. They exceed sometimes predicted no effect concentrations in the case of the traditional environmental endpoints, but also in terms of MICs. And if you tap into MICs, there is this issue of potential hotspots for AMR. And we need to look into this further. So we looked at the same data set, and I want to emphasize how much we can get out from one data set. We looked at the same data set, and we tried to understand how we as a community contribute to the treatment, to the catchment. And here we have population size, a key driver of CECs burden in the receiving environment. We have various groups of pharmaceuticals, lifestyle chemicals, pesticides, illicits, industrial chemicals, and they all pulled together, so different colors indicate different groups. And city A upstream is here, so there is very little pollution here, and city after city D, it's here. We have much more pollution, so daily loads of this various different chemical groups, simply because more population is contributing to this. But what was really interesting to see after doing all the calculations, the population normalized loads show little variation across the catchment. So if you see there's more of a chemical here, simply because there are more of us in this particular area. So it's interesting to see that each of us in this catchment is producing about 154 milligrams of CECs per day. And you can see the variability is very low, it's plus minus 12 milligrams when you look across the catchment. And it's very important information in the context of One Health. If we are the driver, maybe instead of investing in treatment technologies, we should be looking into the way we live and the chemicals we consume on a daily basis. So this kind of at-source treatment solutions might be more applicable to solving pollution problems than end-of-pipe treatment, which is very expensive, and also in the context of climate change, the energy-intensive chemistry and biology treatment processes might not be the way forward. And I can't believe I say this because I'm a chemist and I spent all my PhD trying to develop advanced oxidation processes via catalysis and ozonation, a very energy intensive process. So What we have done, and here there's one example, antibiotics and antibiotic resistance genes. We looked at 3 variables in the system. So one is antibiotics themselves and the metabolites, ARGs, so resistance genes, and also population size. And here we have 5 cities, exactly the same cities that I've discussed, and we have daily loads. These are antibiotics, and this has corresponding resistance genes. And here we have population contributing to the sample. So you clearly see that the highest population, the higher the load, both for antibiotics and resistance genes. The second highest population is here, city C, and this is our Bath city. We have second highest daily load and second highest daily load for ARGs, so both ABs and ARGs. And we can go on. This is the third highest city in terms of population size, and the third highest daily load for both antibiotics and resistance genes. And this shows us very clearly that the population size is the driver here. And when we start correlating antibiotics with population, and here we have Pearson coefficient, we get very high numbers. So this shows very clearly that the correlation is there. There's a linear relationship between population and the usage of pharmaceuticals, and also resulting resistance genes. And I will show you another slide with lots of numbers indicating just that. But I wanted you to look at this. Once we do double normalization, so this is singular normalization population to flows of wastewater. Here we have double normalization population size. This numbers plateau. And we see some very interesting results. So the consumption of pharmaceuticals, antibiotics in this case, per head is similar, and actually similar in this city A and B, and slightly different in city C, D, and E. And it's interesting that they are in 2 different regions. So this is Bath and Bristol region, and this is in Wiltshire. Interesting. Maybe there are differences in the way the prescription happens, but here we are with the results, and the same happens with resistance genes. Now, this again carries very, very important information in the context of how we take this forward. So if we consume pharmaceuticals, antibiotics in this case, and this is linked directly with how many of us there are in the catchment and with the prevalence of resistance genes, the One Health thinking in terms of reduction in antibiotic usage in both in primary, secondary care should be the way forward. And we now have this evidence tool that can capture the issue and also capture whether some of the One Health interventions are successful. And we're trying to do this in our 2 projects. So one is Entrust, and currently working on IPC, Innovative Pathway Control projects, which is Acquia project, looking at various different social and green prescribing and dimension, also educational campaigns. And we work closely with social scientists, and the social science dimension is led by Professor Julie Barnett. She's a psychologist at the University of Bath. Yes, the one dimension that I have not captured as yet, and I will do now, is that the educational dimension, and the fact that we as a society know very little what we discharged via wastewater. And I think most of us think when we consume pharmaceutical excrete, we don't excrete it, it disappears. And also if we dump pharmaceutical down the drain, it's not a big issue because it's 1 or 2 pills. It actually is a big issue because many pharmaceuticals, when they're discarded down the drain, unmetabolized, that they might be causing serious problems down the line. And I want to show you one example, carbamazepine, how we managed to capture direct disposal of pharmaceuticals via wastewater-based epidemiology. So you see here carbamazepine, it's a parent compound, it's an anti-epileptic, and we capture it in one city, city A, over 7 days. And you see very nice, these are daily loads, the black color, and we have also daily loads of its main metabolite, carbamazepine 1011 epoxide. And the red lines indicate carbamazepine intake. So we calculated intake of carbamazepine using its metabolite or parent compound and metabolism pathways. And this is prescription within the catchment. And you can see when you look at carbamazepine 10 epoxide calculations, our wastewater data nicely correlate with prescription in the catchment. And we see that the levels of the biomarker remain constant throughout the week. And we would expect this for carbamazepine because it's a drug that has to be taken over long periods of time. We have seen the spike on Sunday of carbamazepine, the parent compound. If we didn't have metabolite in our study, we wouldn't know that this spike indicates direct disposal as opposed to sudden increase in consumption. And you see on the side, we also spotted direct disposal of various other pharmaceuticals. And this is in 1 week study across the catchment. We managed to capture so many of them being directly disposed. And again, now we have this direct evidence showing that direct disposal happens. If it happens, we need to introduce One Health strategies in the removal of the problem via, for example, intervention strategies that are education based. So, for example, we should be promoting taking pharmaceuticals back to pharmacies instead of throwing them down the drain, as we are doing at the moment. So, just to emphasize this evidence tool dimension. And in this particular case, we have managed to capture disposal of 253 pills at one particular go. And we also seen a similar quantity of fluoxetine, Prozac in our previous sampling campaigns. And this has consequences for the environment because we see this extra loads getting into our receiving waters. So we can do much more with wastewater than just to see where the disposal happens. And this is just a snapshot of what we can do if we look at pharmaceutical usage across the catchment. So it's a very busy slide, and I won't spend too much time on this, but I'll try to explain. So we have 5 cities here. So city A here, city B, city C, D, and E. And this is the population size provided here for you. And we have different groups of pharmaceuticals and also illicit drugs and some industrial chemicals. And here we have population normalized load, so double normalization to flow of wastewater and to the size of population. So if we as a population consume the same across the board, so our behavioral patterns are not different, we should see the same. As you can see, we don't see the same. Sometimes we see less of one group of pharmaceuticals, sometimes we see more. But what's interesting, and these are daily loads, population normalized loads. So in communities, we tend to behave in the same way for diabetics, for example, but we have differences in terms of comparison of various different cities, so intercity comparison. So intracity is similar, but intercity is different. And this is really interesting because we can use pharmaceuticals as a proxy for health. So, for example, in the city C, we have lower usage of antidiabetics. And this is my very loose interpretation. We are trying to triangulate this data now with socioeconomic indices and public health records. So please don't hold me to what I'm saying at the moment. Just want to show what the approach can provide us with in terms of information. So lower level of antidiabetics in this particular city might be linked with the fact that this city actually has the lowest deprivation index in this particular catchment. Now, here we have, in city E, we have the lowest usage of cardiovascular pharmaceuticals, but the highest usage of illicit drugs. And you can actually see the spike of illicit drug usage during weekends. So there is this temporal dimension here in what we can capture. And it might be because this is the largest city in the catchment and actually is the city with the youngest population. Here we have 2 cities with very high levels of some industrial chemicals. So city B and city E. And actually these 2 cities have about 30% industrial contribution, if you remember this orange color on the first on the map that I showed you. And it might be that it's because of the industry. The population living in these two cities has high exposure to certain chemicals, as opposed to other communities that don't have this industrial presence. So very important information can be drawn from this kind of data set. And actually, if we have limited resources, again, me thinking about One Health, we might think about, okay, this community requires a bit more work, maybe much more high resolution testing than other community. We have also looked at how various different markers behave across the board in terms of various different cities. So if we see them the same across, this is population normalized load, and I'm conscious of time. I think I have to finish very soon. We have population normalized load here, and if the various different markers behave the same, that means that the usage across the board is constant. So the population is the one that drives the presence of markers, and we have changes in various different cities. For example, we have increase in nicotine here usage, or illicit drugs usage, or industrial chemicals usage. This means that the levels of markers in wastewater might be linked with cities' function. So this graph, you might not see this very clearly because I don't have time to explain it well. This graph shows that some markers that we capture in our studies will be directly linked with population size, such as this one here, so many pharmaceuticals. Other markers will be linked with the exposure that we have due to cities' functions. So, for example, presence of industry or lifestyle choices. And I pulled it all together in 3 groups. And we've done 2 things here. And I'm not going to get into details because there's no time, but I wanted to emphasize that some pharmaceuticals are really linking very nicely with population size and link nicely with the linear regression model. So there is this clear correlation between population size and the levels of biomarkers, and this is represented by R squared and an R value. We also looked at p value to identify whether there is a clear relationship, positive relationship between various different markers and population size. And we have this first group of chemicals, so analgesics, antidepressants, antidiabetics, so high usage chemicals, lifestyle chemicals, anti-inflammatories. And we have clear correlation between population size and the usage of this chemicals. And they can be actually used as a population size markers. Now, second group of pharmaceuticals and other chemicals, such as water quality indicators, do not correlate as well, though correlation is still visible. And the third one has the lowest correlations. And these are mainly industrial chemicals. These are antihistamines and also anti-cancer drugs. So the chemicals that might be used, pharmaceuticals that might be used seasonally. And also pesticides, and I mentioned industrial chemicals, then they will be very much linked with the geographic location, for example, of some industry. And these are key chemicals markers that we should be using to estimate, to understand where possible problems and issues are in terms of early warning. So very briefly, just 2 slides in trying to point out what we have managed to capture, again, in the same study, in the same data set. And we've been digging in, looking into this data set for several years now. So bisphenol A. Bisphenol A is well-known chemical that is used in plastics, and we expose ourselves to bisphenol A via food consumption. So it's in, for example, in fast food, in packaging. If you like fast food, if you microwave it with packaging on, you are exposing yourself to bisphenol A. And Food Safety Authority says that this is our main exposure. There's no exposure via other routes. And exposure is at low level. So what we wanted to do, we wanted to see whether this is happening in our catchment. So we looked at 5 cities, And you can see that city A, C, and D, and these are the cities that have only communal discharge, have very low exposure levels. So alongside food safety authorities standards, but you can see city B and E have really high levels and in this case, we are looking at bisphenol A sulfate. It's not the parent compound, it's the indigenously formed metabolite. So this is our biomarker. And we have high levels in these two communities, B and E, where there is industry. And industry here is very much linked with food packaging and with paint formulation, so high exposure to bisphenol A. And we see this also in a way that we see increased levels during weekdays versus weekends. You can see weekend exposure is very low. And this makes me think that this is occupational exposure, something that we'll not necessarily be able to capture by our other routes. And this slide, again, very quickly, this is bisphenol A again, but we compared this with pyrethroid pesticide intake. And pyrethroid pesticides, we expose ourselves to this pesticide again via food, and we see this exposure. And interestingly enough, and I always say it as an anecdote, maybe more than anything else, we have this increased intake of pyrethroid pesticide during weekend. And I don't know what is the reason, but one of the reasons that I usually provide is that during weekends we cook more at homes, we eat more vegetables because we want to have better lifestyle, and maybe as a result we're exposing ourselves to pyrethroid pesticides. So this is my final slide, apart from acknowledgements, and this is how our results look like. So we do wastewater-based chemical mining, and also we mine for biologicals, and we look for various different chemicals together. I showed you very few examples, but we have hundreds of compounds that we look at. And the key to success is to know what to do with the data, to model it and understand what it does. And you can see 4 chemicals captured here. So the indigenously formed chemicals and the stressors themselves. So we have here pyrethroids, pesticides, BPA, cocaine, and this is fluoxetine, so known as Prozac, antidepressants, and the biomarkers. And we see how our exposure to them changes. Some remain constant throughout the week, some increase during weekends, and some decrease during weekends. And it can give us really powerful information about, our health and lifestyle full stop. So I will leave it there and just acknowledge everyone who's been involved in this study, other studies that led to this work. So I have already mentioned our excellent environmental chemistry public health research group working so hard to get the data. And Wessex Water, we wouldn't be able to do this data without Wessex, produce this data without Wessex Water. And the support throughout years and an incredible contribution also in terms of driving some of the activities, funding some of the activities, providing data to crunch. And networks across the board, so EU and also international networks, and our funders, so European Commission, UKI, and charities and industrial partners. I will leave it there. Thank you very much. Apologies if I've been talking for too long. As you have heard, I'm very passionate about this work. So I'll stop talking for now.
Thank you, Barbara. It's very impressive. A lot of things we can learn from you.